Happy Saturday, scientists. Let's review. We're going to talk through the B6 Part 2 revision worksheet in this video, and if you have any questions, please ask those in the comments below. You'll find a link to the detailed answer key in the description of the video. It's important that you understand the difference between pathogens, the things that generate the pathology of a disease, and vectors, the things that allow the pathogens to move from one organism to another. That vector can be another organism sometimes, so for example mosquitoes, which carry the protozoa that causes malaria, or they can be something like water, which allows bacteria to move from person to person in the contaminated water, and so if another person drinks that contaminated water, they can become ill. When we're looking at bacterial replications, we are looking at one potential cell, or potentially more than one, and it will have its plasmids and other genetic material in it. The bacteria will be able to split itself into two after copying its genetic material. So we'll have two bacterial cells with an identical copy of the genetic makeup, and each of those is then able to split into two more cells. And each of those again into two more. When we're discussing replication of viruses, we have two things that are involved. First of all, we have the virus itself with its genetic material, but we also need some form of host cell. The virus will have to attach itself to the host cell and inject its DNA into that host cell. The virus will then hijack the host cell and use it to copy the genetic material of the virus. Once it has done that, it will instruct the host cell to manufacture and assemble new viruses. And eventually, the host cell will burst open and those viruses will be released into the surrounding tissue, and or they could be coughed out by a person or sneezed out by a person and go on to infect other people and other cells. We have ways that we can prevent these. All of these are logical, and lots of these are being discussed in 2020 and 2021 in terms of covering your nose and mouth. Also logical things such as not touching infected people or objects, using protection during sexual intercourse, not sharing needles, washing hands, and cooking food properly are all going to contribute depending on what the pathogen is. You need to know about your body's defensive barriers and how those defensive barriers are able to ensure that your body isn't vulnerable to infection, or is at least less vulnerable to infection. When your immune system is going to respond, you have a simplified view of this that you need at GCSE. It depends essentially on two categories of cells, the phagocytes and the lymphocytes. The phagocytes are going to capture and consume pathogens, breaking them down, and then using those parts for identification. So the lymphocytes will then recognize the pathogens that the phagocytes have broken down and produce the appropriate antibody or antitoxin for that pathogen, and then retain that memory so that if there is a future infection, their response is much more rapid and much more correct or targeted. And this is the process that vaccination will take advantage of. You give someone a weakened part or a weakened virus, and they are then able to start to prepare that immune response. And if there is a detection of that virus, or if there isn't a future infection of that virus, the immune system is already pre-learnt how to deal with that virus, and it therefore already has some antibodies in the blood and is able to react much more rapidly. We know that there are certain pathogens that can be targeted with specific agents, such as antiseptics, antivirals, antifungals, or antibiotics, but you need to use them appropriately and for the appropriate pathogen. So for example, giving someone an antibiotic if they have a viral infection is completely useless because it's not going to have any effect on the virus. The way that we go about testing the effectiveness of these antibiotics is one of your required practicals. So hopefully you remember where you plated out some bacteria on an agar plate, and then on top of that dish you added sterile filter paper discs that were either done with a different dose of the antibiotic or with different antibiotics to see which one is most effective on this bacteria. Once the bacteria are allowed to grow, there will be zones of death, or zones where the bacteria are not able to grow, around each of the effective antibiotics, And if there's no effect, then the bacteria will grow right up to the size of the filter paper disc. So if we looked at that disc, it would look something like this. All the blue here is the bacteria, and then we have these areas of clear zone. We can then go about measuring the diameter of the area, and we do this in two different directions and take an average, and this deals with any irregularities in the shape of the clear zone. There is an error in this answer key here. Because the diameter here is 2 centimeters, the radius is actually in fact 1 centimeter. And so therefore this should be 1 squared times pi, which is 1 pi, or 3.14 centimeters squared. I would like to pretend that I had done this on purpose because it is a common mistake that students make. However, this is also apparently a mistake that teachers can make as well. This has been corrected in the answer key that's linked in the description. And of course, you now have the correct answer on screen as well. You need to understand the general process that we go through when we are trying to develop new medical treatments or drugs. We begin with computer models that will test a large number of possible compounds. Once that is being shrunk down to a few, or possibly none, those few can be then tested on organisms and tissue samples in a lab to determine that they don't have any obvious toxic effects. It will then be tested on healthy individuals to assume that that there aren't any harmful side effects. And so therefore, because the person is healthy and doesn't have the condition, taking this drug should have no effect on them. Once this has done, we can go on to test if the drug is actually effective against the illness. And so we'll find volunteers that have the illness and give them this treatment. This will then be done in a larger scale if it has proven initially effective, and usually this is done in a double blind in order to minimize the effect of placebos. There are a number of different factors that can increase your risk of cardiovascular disease, and these center a lot around lifestyle factors, but there are also some genetic diseases that will be included in this section. At question 19, remember that there are other treatments as well that are coming into this new cutting edge area of medicine. So you could be asked about some of those other treatments in terms of their moral and ethical implications as well. I hope that you found this worksheet useful. If you do have any questions, please leave those in the comments below. In the description, you'll find a link to the detailed answer key for this worksheet. And until then, happy sciencing.